Hello everyone, this is the chapter 10 of level 2. In this chapter, we will discuss about nitrogen metabolism, molecular nitrogen, nitrogen fixation, nitrate and ammonia assimilation by plants, amino acid synthesis. So nitrogen metabolism, all the living organisms are basically composed of carbon, hydrogen, oxygen, nitrogen and many other forms of chemical elements. These elements contribute to finally organize various biomolecules present in a cell. Nitrogen is next to carbon in importance in living things. In a living cell, nitrogen is an important constituent of amino acid, protein, enzymes, vitamins, alkaloids, and some growth hormones. Therefore, study of nitrogen metabolism is absolutely essential because the entire life process is dependent on these nitrogen containing molecules. In this lesson, you will learn about various aspects of nitrogen metabolism, including nitrogen fixation and nitrogen assimilation in plants. After completing this lesson, you will be able to describe the modes of nitrogen fixation, explain the steps involved in nitrogen fixation by free living organisms, explain the mode of symbiotic nitrogen fixation in leguminous plants, describe the assimilation of nitrate and ammonia by plants, describe amino synthesis in plants.
Nitrogen
Now you can see
fixation.
this animation.
Objective: to learn the process and importance of nitrogen fixation in plants. Nitrogen is the most important component in living organisms in the form of DNA, RNA, and proteins. Our atmosphere consists of about 78% nitrogen. Although atmospheric nitrogen is not in a form that is usable for most living beings, they are able to use it when it takes the form of compounds. Atmospheric nitrogen can be converted into nitrogen compounds during a process known as nitrogen fixation. The nitrogen from the atmosphere precipitates and is deposited into the soil and water. Once in the soil, the nitrogen will combine with hydrogen to form ammonium ions through the aid of microorganisms. This process of converting the atmospheric nitrogen into nitrogen compounds by microorganisms is known as nitrogen fixation. One of the best known examples of nitrogen fixation is the association between leguminous plants and bacteria of the genus Rhizobium. Leguminous plants, such as beans, have these bacteria within the nodules of their roots. The bacteria first enter an epidermal cell of the root, causing the root to develop an infection thread. The bacteria will then move through the infection thread until it reaches a cell deep within the cortex. The cortex cells will divide quickly, forming a nodule. The rhizobia will also multiply rapidly within the nodule. The bacteria, now referred to as bacteroids, will begin the conversion of nitrogen to ammonia. Once atmospheric air reaches the root nodule, oxygen will combine with the leg hemoglobin. Leg hemoglobin combines with oxygen, thereby promoting nitrogenase activity. The nitrogen molecules, with the help of nitrogenase, will combine with hydrogen molecules to first form ammonia and then an ammonium ion. Ammonium ions will be utilized by the plant as long as it lives. After plants die, the nitrogen will still remain in the soil where it will enrich it. Summary: nitrogen accounts for about 78% of the atmosphere. In nature, atmospheric nitrogen is converted to nitrogen compounds with the aid of certain bacteria in a process known as nitrogen fixation. Rhizobium bacteria fix nitrogen in leguminous plants.
Molecular nitrogen. Nitrogen is primarily present in the atmosphere freely as dinitrogen or nitrogen gas. It is present in the combined form as Chile saltpeter or sodium nitrate, and Chile in South America is the major source of this nitrate nitrogen. Molecular nitrogen or diatomic nitrogen is highly stable as it is triple bonded. Because of this stability, molecular nitrogen as such is not very reactive in the atmosphere under normal conditions. In the atmosphere, molecular nitrogen is 78.03% by volume, and it has a very low boiling point, minus 195.8 degree, which is even lower than oxygen. Proteins present in living organisms contain about 60 nitrogen. So nitrogen fixation, the conversion of molecular nitrogen into compounds of nitrogen, especially ammonia, is called nitrogen fixation. Nitrogen fixation is a reductive process. Nitrogen fixation will stop if there is no reducing condition or if oxygen is present. This nitrogen fixation may takes place by two different methods, abiological and biological. Abiological nitrogen fixation, the nitrogen is reduced to ammonia without involving any living cell. Abiological fixation can be of two types, industrial and natural. For example, in the Haber's process, synthetic ammonia is produced by passing a mixture of nitrogen and hydrogen through a bed of catalyst iron oxide at a very high temperature, and you can see this in reaction. This is industrial fixation and nitrogen reduced to ammonia. In natural process, nitrogen can be fixed especially during electrical discharges in the atmosphere. It may occur during lightning storms, and nitrogen in the atmosphere can combine with oxygen to form oxides of nitrogen. These oxides of nitrogen may be hydrated and trickle down to earth as combined nitrite to nitrate. Biological nitrogen fixation. Chemically, this process is same as abiological. Biological nitrogen fixation is reduction of molecular nitrogen to ammonia by a living cell in the presence of an enzyme. So nitrogen fixation by free living organisms and symbiotic. So nitrogen fixation is a distinctive property possessed by a special group because of the presence of enzyme nitrogenase in them. So the process of nitrogen fixation is primarily confined to microbial cells like bacteria and cyanobacteria. These microorganisms may be independent and free living. You can see them in the next table. Some free living microbes which here you can see in this table. Clostridium, this is non-photosynthetic. Klebsiella, Azotobacter, Rhodospirillum, this is photosynthetic. Anabina, this is also synthetic. So some microbes may become associated with other organisms and fix nitrogen. The host organism may be a lower plant or higher plant. The host organism and the nitrogen fixing microbes establish a special relationship. This is called symbiosis, and result in symbiotic nitrogen fixation. You can see in the next table. System and symbionts like lichens, association between algae and fungi, bryophytes, pteridophytes, gymnosperm, angiosperm, again angio, next one is angio. So these are all. So this is the mechanism of biological fixation of nitrogen. It requires the molecular nitrogen, a strong reducing power to reduce nitrogen like FAD, flavin adenine dinucleotide, a source of energy to transfer hydrogen atoms to dinitrogen, an enzyme, nitrogenase compound, for trapping the ammonia formed since it is toxic to cells. The reducing agent and ATP are provided by photosynthesis and respiration. The overall biochemical process involves stepwise reduction of nitrogen to ammonia. The enzyme nitrogenase, it contains protein and binds with molecules of nitrogen at its binding sites. This molecule of nitrogen is then acted upon by hydrogen and reduced in a stepwise manner. It first, it first produces diamide, then hydrazine, and finally ammonia. Ammonia is not liberated by the nitrogen fixers. It is toxic to the cell, and therefore these fixers combine ammonia with organic acids in the cell and form. So molecular nitrogen is a very stable molecule. Therefore, sufficient amount of cell energy in the form of ATP is required for stepwise reduction of nitrogen to ammonia. In legumes, nitrogen fixation occurs in specialized bodies called nodules. The nodules develop due to interaction between the bacterial rhizobium and the leguminous roots. You can see this in the next diagram. The biochemical steps for nitrogen fixation are same. However, legume nodules possess a special protein called leg hemoglobin. The synthesis of leg hemoglobin is the result of symbiosis because neither bacteria alone nor legume plant alone possess the protein. Recently, it has been shown that a number of host genes are involved to achieve this. In addition to leg hemoglobin, a group of proteins called nodulins are also synthesized, which help in establishing symbiosis and maintaining nodule functioning. Leg hemoglobin is produced as a result of interaction between the bacterium and legume roots. Apparently, rhizobium gene codes for heme part and legume root cell gene codes for globin moiety. Both the coded products together constitute the final protein leg hemoglobin. So this is complete chart. Host plant cytoplasm and rhizobium in the nodules. Nitrate and ammonia assimilation by plants. As pointed in the previous section, nitrogen fixation is confined to selected microbes and plants, but all plants do require nitrogen because it has a role to play in the general metabolism. So therefore, plants which do not fix nitrogen use other combined nitrogenous sources such as nitrate and ammonia for carrying on metabolic activity. Nitrate is absorbed by most plants and reduced to ammonia with the help of two different enzymes. The first step conversion of nitrate to nitrite is catalyzed by an enzyme called nitrate reductase. This enzyme has several other important constituents including FAD, cytochrome, NADPH or NADH and molybdenum. The overall process of nitrate reduction takes place in the cytosol and is an energy dependent reaction. The enzyme nitrate reductase has been studied in many plants and it is observed that the enzyme is continuously and synthesized and degraded. The enzyme nitrate reductase is inducible. This means that increase in nitrate concentration in the cytosol induces more of nitrate reductase to be synthesized. However, when excess in plants, it has also been observed that light also increase nitrate reductase when nitrate is available. In the second step, the nitrite so formed is further reduced to ammonia and this is catalyzed by enzyme nitrite reductase. Nitrite present in the cytosol is transported into chloroplast or plastids where it is reduced to ammonia. The enzyme nitrite reductase is able to accept electrons from sources such as NADH, NADPH or FADH2. Besides, reduced ferredoxin has also been shown to provide electrons to nitrite reductase for reducing nitrite to ammonia. Ammonia so formed has to be utilized quickly by plants because accumulation of ammonia has a toxic effect. Some plants including algae leach out excess ammonia which can further be oxidized to nitrite and nitrate by microorganisms in the soil or water. Amino acid synthesis by plants. As you have noticed that ammonia formation is achieved by plants either by nitrogen fixation or by reduction of nitrate to nitrite. Ammonia is the most reduced form of inorganic combined nitrogen. This ammonium now becomes the major source for the production of amino acids, which are the building blocks of enzymes and proteins. Amino acids have two important chemical groups, amino group and carboxylic group. A typical amino acid with functional group R represent alkyl group. Ammonium produced is the major source of amino group. However, the carboxylic group has to be provided by other organic molecules synthesized by the plants. There are two major reactions for amino acid biosynthesis. Reductive amination reaction. In this reaction, ammonia combines with a keto acid. The most important keto acid is the alpha keto glutaric acid produced during the operation of Krebs cycle. You can see this in the chapter 12 of level 2 respiration. The keto acid then undergoes enzymatic reductive amination to produce an amino acid. Similarly, another amino acid called aspartic acid is produced by reductive amination of oxaloacetic acid. It has been noted that reductive amination represents the major port of entry for ammonia into the metabolic stream in plants. This initiates synthesis of glutamic acid followed by other amino acids. Transamination. So this is another very important reaction for amino acid biosynthesis. The reaction involves transfer of amino group from already synthesized amino acid to the keto acid. You can see this in this reaction. In the above reaction, aspartic acid has transferred its amino group to the alpha ketoglutarate acid to synthesize glutamic acid and release keto acid. The reaction is catalyzed by enzyme. This is called transaminase. A large number of amino acids are synthesized by this transamination reaction. Amino acids are organic molecules containing nitrogen. The incorporation of amino group from ammonium into acid represents the major steps for synthesis of nitrogenous organic molecules. So what you have learned in this PPT, nitrogen is an important constituent of several biomolecules such as amino acids, proteins and enzymes. Molecules such as vitamins, alkaloids, nucleic acids, pigments and some growth hormones also contain nitrogen. Molecular nitrogen is triple bonded and stable. Nitrogen fixation is the reduction of nitrogen to ammonia. Abiological nitrogen fixation is an industrial process. This is also called Haber's process. Biological nitrogen fixation takes place in a living cell. The enzyme that catalyzes nitrogen fixation is nitrogenase. Nitrogen fixation may take place in free living organisms or in symbiotic systems. There are many symbiotic nitrogen fixation systems such as lichens, pteridophytes, bryophytes, gymnosperms and legumes. Cyanobacteria is the symbiotic component in lichens, bryophyte, pteridophyte and gymnosperms. In legumes, the symbiont is a species of bacterium, rhizobium. Source of electrons and energy for nitrogen fixation is generally acid. Hydrogen gas evolution may also accompany nitrogen fixation process. Nitrate is the most oxidized form and ammonium is the most reduced form of nitrogen. Nitrate is reduced to nitrite by an enzyme nitrate reductase. Amino acids have two functional groups including amino group and carboxylic group. Amino acids may be produced by reductive amination of keto acids. Amino acids may be produced by transamination reaction. Reductive amination reactions are catalyzed by dehydrogenase. Transamination are catalyzed by. So these are the questions for our practice. Thanks for being with me. If you like this study material or this PPT and animation, please subscribe this channel and forward to all also. We will meet in our next chapter. Thank you very much.